Good morning. It's 9 a.m. here in San Diego, so we'll begin today's webinar. Today's webinar is on the subject of predicting the effects of mutation, on protein stability, protein-protein interactions, peptide binding, and small molecule drug binding. Thank you very much for joining us. If you have any questions during the webinar, please use the question panel, or at the end of the webinar you can raise your hand and I can open your mic. My name is Andrew Ori, and if you have any questions after the webinar, please feel free to email or call, and I'll be happy to answer your questions. I think most of you already have the software downloaded, but if you don't have it, you can just go to our support site, download ICM Pro, the full package, and send me the host ID, and then I'll send you a license key to activate the software. And it runs on Windows, Mac, and Linux. So this is the second to last webinar in the series. The final one is next week on the subject of ligand-guided modeling, how to use a ligand and binding information of a series of binders in order to be able to model your binding pocket and mold the pocket according to known binders. This is the last one in the modeling series. We're also holding a workshop, a one-day workshop in San Diego if anyone is around, on April 16th. It's a free workshop. It covers protein modeling as well as drug design and lead optimization. If you're interested in the other webinars in the series, you can go to YouTube and just search for MuleSoft, and you will find a number of different webinars and tutorials on different subjects in ICM, such as docking and other modeling tools. If you're not familiar with MuleSoft, MuleSoft was founded by Ruben Abagayan, and our principal scientist is Max Totroff, and our principal developer is Eugene Rausch. And if you're wondering what ICM stands for, it stands for Internal Coordinate Mechanics, which is the coordinates that we use rather than Cartesian. We use internal coordinates. And you can read the original papers from Ruben and Max from 1994, which describe this method, as well as the optimization method, the bias probability Monte Carlo method that is used for predicting the effect of mutations and modeling and docking and other tools. So today's webinar topics, we're going to start with sort of a general modeling question, just how to mutate a residue in a protein and optimize the vicinity of the region. That's not necessarily predicting the effect of the mutation, it's more just making a model with a mutation involved. And then we'll be moving on to how to predict the effect of a mutation, predict the effect of mutation on stability, protein binding, peptide binding, small molecule binding, as well as how to scan a peptide for different mutations when it's bound to a protein receptor, and also how to scan the pocket for stabilizing mutations as well for ligand, for small molecule binding. So applications. So obviously wild type protein function is directly related to the folding free energy, and this is obviously characteristic of protein stability. So changes in protein stability can be due to missense mutations or naturally occurring mutations, and commonly in the experimental lab people are making point mutations in vitro to understand the contribution of a particular amino acid to the stability of a protein. So obviously predicting the effect of a mutation can provide useful information for experimental design, protein engineering, and understanding molecular mechanisms. I'm just going to start with just a very recent paper that's come out, just found using ICM. It describes describes some functionally relevant mutation sites in the human mitochondrial respiratory complex. And just to show you what kind of things you can do in ICM, as part of the paper they made a fully interactive molecular document, which you can see here. It's quite useful because they have a table here with all the different mutations and a possible explanation of the mutation. So if we're interested in looking at this alanine to threonine mutation, we can just click on this link and it takes us directly to that alanine residue and the threonine. So in this paper, they've basically used ICM to make mutations. They're not necessarily predicting the effect of that mutation, but they're taking a crystal structure and they build a model with that mutation included. So we're going to see how to do that in a little bit as well. So you can use these. This is using MolSoft's JavaScript program software, which is free, and basically it's a series of slides, and you can just if you go to MolSoft ICM, you can just make some slides, and then you can export it to HTML, which we're showing here. This is the web browser I'm showing. So it's just a one example. Another application which is particularly useful. Just wanted to share a paper from the Catridge lab at USC. So they used ICM and some other methods for protein engineering of G protein coupled receptors for the computational design of thermostabilizing point mutations. So the paper is in eLife in 2018. So obviously for most GPCRs, thermostabilization needs to be used for the successful structure determination of GPCRs and other biophysical studies. So this method, they call it CompoMug, and it basically uses a sequence-based analysis and structural information and some machine learning to predict mutations for thermostabilizing this GPCRs. It was tested experimentally on the serotonin GPCR, and the predicted mutations enabled crystallization and structure determination of the 5-HT2C GPCR receptor in both the active and inactive states. So the computational modeling helped the experimental scientists to crystallize the structure, basically. So how can we make a mutation in ICM? So firstly, we need to read in this protein structure from the PDB. We need to convert it from a PDB structure to an ICM object, and then we basically right-click on the residue, and then we can mutate. So I'm going to show you how to do that in ICM. So if you have ICM downloaded, you should have a icon you can click to load the software. And I'm just going to use an example with the PDB code 1TP5. Hopefully you can see. So you go to the search tab, search 1TP5. And this is a PDZ domain, which plays a role in anchoring receptor proteins to the membrane and other cytoskeletal components as well. So we have this PDZ domain with a peptide bound shown in yellow. You can see 2 molecules of water molecules as well. So to make a mutation, we need to first convert this PDB structure into an ICM object. So you right-click on the name of the protein in the ICM workspace, and you click Convert PDB, and that will convert it to an ICM object. So there's some options. We talked about these before in previous webinars. If you're doing this properly, you would optimize all the hydrogens. It takes longer. Optimize asparagines and glutamines, for example, from the X-ray structure in order to get the correct orientation in terms of the hydrogen bonding network and the most energetically favorable orientations. I'm just going to replace the original and just go OK. And that will convert. So now you can see in the ICM workspace it says ICM here, which means it's an ICM object. It's not an X-ray structure. So it has hydrogens added. it's fully optimized for modeling and docking and prediction of mutations. But in this example, we're just making a mutation. We're modeling a mutation. Okay, so it'd be nice to see how this peptide interacts with the protein. So the peptide is the yellow molecule. One quick and easy way to do that is to right-click on the ICM workspace here, choose shapes, choose molecular shapes and contacts here, and it will build some surfaces so we can see. So it basically builds a surface of the protein and of the peptide shown in yellow. I'm just going to take the peptide surface away, and then we're left with the contact. You can also see that the contacts on the protein surface are colored as well, yellow, and it highlights. And so we can see in this this peptide makes it's kind of a slightly buried pocket here with some hydrophobic interactions. So we can play around with some different amino acids at this point. We can make a mutation. For example, for this valine, to make a mutation, we're just going to right-click and choose Advanced, and you can choose Mutate Amino Acid. And then you can then choose the amino acid from the list here. So you can just put an arginine there. You can see we probably have some clash with the protein, but this is just an example. So we're not predicting the effect of this mutation. We're just modeling that effect. So then once you've made your mutation, you can right-click and you can choose Advanced, and you can choose Optimize Side Chains, and then you can see the side chain dancing around trying to find a way to fit into that pocket. It's probably not going to find a decent orientation because it's too large really to fit into that region, but it's just an example. So you'd make the mutation, and then you will run this optimization. You can also select multiple residues on the peptide as well as on the protein to model that region. So you can also mutate, right-click and mutate on the residue, choose Advanced again. You can attach a glycan sugar there, or you can. We also have a list of sort of unnatural, non-protogenic amino acids as well, which you can mutate too. So you just need the list here. Okay, so if you wanted to also include when you're optimizing it, you can also include the selection here and model that region. It's going to return it back to its original residue, which was a valine, I think, and okay, so that's how to make a model of mutation. If you have multiple mutations in the protein, you're probably better off using, rather than doing interactively like this, you can make a model, a homology model, and you can look back to our previous webinars on how to do that. You just make a sequence alignment with your mutations, and then that would build a full model with those mutations. But this is just for like point mutations, visualization, and also you can evaluate how energetically favorable that mutation is. Okay, so our first, so now moving away from modeling, but basically onto prediction now of the effect of a mutation. So in this example, that we have a method for evaluating the protein stability. So this method computes the change in protein stability upon mutation of a single residue. a PDB structure or ICM object containing the protein is required, and the free energy change in protein stability is computed as follows, like delta G equals the delta G of mutant minus the wild type, and then we compare the folded with the unfolded delta G. So the free energy of the unfolded and misfolded states is approximated by a sum of the specific energies for each residue, and the residue specific energies were derived empirically using a large set of experimental data. The mutation of a given residue is followed by the biased probability Monte Carlo simulation with flexible side chains for the residue that you mutated, as well as the neighboring residues. And the rest of the protein is considered to be rigid for this. So this is prediction of protein stability. So we have one example here, which has the PDB code 1HB6, and it's a good example because there's a lot of experimental data. If you look up this, it's a structure of a bovine acyl-CoA binding protein, and then there's lots of mutation data derived for this molecule. So you can compare the predictions. So we go back to ICM for this example. It's going to go. It's going to delete everything it did here. And so for this example, the code is 1HB6. Shown here. Okay, so we have a cadmium ion here, which is just on display. And we have some residues shown by default. We can just undisplay those as well. And so to make the prediction of stability, one residue in particular that, if you look in the papers for this structure, there is a lysine, I think. Sorry, phenylalanine 26. So to find phenylalanine 26 in this structure, we can expand here in the ICM workspace, and we can make a selection. So number 26 would be this phenylalanine here. Okay, so we just select that. If you select it, click and drag over the F, you can see that that selection is propagated to the 3D structure here. So we can just display the stick, and that side chain is displayed. So we're going to try and evaluate the delta G for on protein stability for this mutation from phenylalanine to an alanine residue. So to do that, we go to the mole mechanics menu shown here at the top, and then there's the option called try mutation. So all the predicting the effect of mutation options are in this menu here. So if we expand that, we can see them here. Okay, so we have a few examples. If you just load them, then it explains how to do it. Basically, what I'm showing here, you can go through the steps. But for this example, we're going to use protein stability. So we go to mole mechanics, try mutation, protein stability. And then we have this dialog box shown here. And you can see that we've made one residue selection. It's found the one selection that we made. The green crosses here means a graphical selection. Green crosses here. It matches. And then you can make a variety of amino acid changes. In this example, we're just going to choose alanine, just like this and go OK. And then I'll set it running and I'll come back to this dialog box. So we can see this mutation, this prediction is running. It's got one background job here. And at the bottom you should see the, down here you should see a progress bar for the prediction shown here. So there are other options in this dialog box. We're waiting for this to finish running. So you can check multiple amino acids that you want to try at that position, and you'll get a prediction delta G for each one. Or you can choose all. Now it'll do all the protogenic amino acids. However, if you may be interested in unusual amino acids, therefore we have this extra box here where we have a list of the common unnatural amino acids. So you can just select here and it will try that. You can select more than one example. If the one you're interested in is not listed, then you can draw your unnatural amino acid side chain. So you'd click on this extended list of amino acid button here, then go to draw. There's this, hopefully you can see, just check. There's this button here which will open an editor where you can sketch your amino acids. So a quick way to start would be to go to the templates. So it's basically like a ChemDraw environment where you can sketch. So you could sketch out your example. I'm just going to get the results here. So, or there's some templates. There's actually an amino acid set here, so you can just, this is just a quick way to start. You could choose alanine, and then you can start to sketch whatever unnatural amino acid you want to try at that position. But more than likely, the most common ones are listed here, so you might find it in this list here. And once you've made your change, once you've sketched it, then you can add it to this list for future use. You can also have a selection for backbone chirality. You can change it from L to D if that's what you're interested in, and also this option keep all chains. So if you've got multiple chains in your protein, when it's doing the simulation, it will, by default, it will remove the chains that aren't involved in the prediction. But if you want to keep them, then you just click here. We'll do it. Okay, so you can see this example. There was just one amino acid that I changed. We have it. Once it's finished, you'll get a table like this. It tells you the protein that it's changed, the chain, the residue number that you changed, the wild type, the mutant. You say we changed it from phenylalanine to alanine, and then it has the delta G prediction, and also for solubility as well. So the actually experimental value, the prediction with the experimental value is very close. I think it's like 1 point. I think experimental delta G for binding for the mutation of this residue is I think it's like 1 point. I had it in the previous slide, but you can check. You can also try other mutations as well, but it's very close. So if you had a big table, you can just double click and it will take you to that mutation. So now it's an alanine. And then you can see in the ICM workspace, so you just double click on your table, it's loaded the mutant here. So you now have two objects. You have the original crystal structure that you read in, and also the mutant. So when we expand this, we should see at position 26, I think it was, there's an alanine residue, and it's a phenylalanine in the wild type. Okay, so that's protein stability prediction. Let's have a look at protein-protein interaction. So I'm just going to close this. There you go. So for protein-protein binding or protein-protein interaction, the effect of mutation is a slightly different procedure. So the method computes the change in binding free energy of a protein complex upon mutation of a single residue. The energy is computed as the interaction energy between two parts. So you need to define the part of the protein which has your mutation, and then the other part is the interacting region. So in the example we're going to read, we're making a mutation to one of the molecules, and which actually interacts with two other chains. So we need to include those in the prediction. The binding free energy change, delta G, is computed as a difference between the binding free energy of the mutant and the wild type. The energy is calculated for fixed backbone and all the side chains except those in the vicinity of the mutatable residue. So it basically optimizes the side chains of a sphere around the mutation that you're making. And then a Monte Carlo bias probability Monte Carlo simulation is carried out to relieve any clashes that may have occurred. For example, if you're mutating to a larger amino acid, there may be a clash, so you have an optimization procedure embedded. Okay, so I had a question regarding delta G. So if in the previous example, the delta G was greater than 0, so that's generally thought of as a less stable mutation. If the delta G is less than 0, then you may be increasing the or maintaining the stability of the protein with that mutation. So negative and positive. Okay, so how to do this for protein-protein interaction. So for this example, we have protein structure with, it's going to load ICM again. We have a big protein with PDB code 1 CBW. You go to search, just type 1 CBW. So it's a complex of the Alzheimer's amyloid beta protein precursor complexed with chymotrypsin and trypsin and another inhibitor. This example, the resolution is 2.6 angstroms. And if you read the paper associated with this crystal structure, they have done a lot of phage display experiments to randomly mutate certain residues within the binding region of the chymotrypsin. And so there's a lot of experimental data for prediction of protein-protein interaction, which you can try and compare to the prediction in ICM. So the residue we are interested in, so we can see that this has multiple chains. The coloring is propagated here, so this is H chain here. We have the orange one, which is the D chain. And another question about when we optimize the residue, are the interacting residues optimized as well? Yeah, so if you're making a mutation here and it is close to the other chain, then there's going to be a simulation in this region, so it's going to incorporate residues from both chains when it makes the prediction. So in this example, we're going to look at lysine 15, which is in the D chain here. So if we zoom in, this is lysine 15, which is this residue. So you just click and drag over that residue, and we could center on that region. Just display the center works on any selection, so it's centered on that. But for this protein-protein interaction, it'd be good to understand what other chains interact with this chain. So what kind of protein-protein interactions are going on? One way to do that, we do have actual methods to predict protein-protein interaction sites, but in this case, obviously, we know. So we can just select this chain D, and then we go to we can go to Tools, and we can choose the option Analysis, and then there's Contact Areas. So I just have one selection. I'm just going to choose the option One Selection, and it's asking me the molecular selection. It's just one molecule, and do we want to just basically this option is going to show us where this which proteins this chain interacts with, basically. And we just go OK, and then we'll see that we now have a table which shows the ranks by the residues that make the biggest interaction with our lysine. So lysine is number one, obviously. So this arginine in chain D has a high contact area. And then if we go down the table, you can see that other chains also make. Chain I makes an interaction, chain C, but we're mainly interested in the interaction in the vicinity of the lysine. So the lysine is here, and the side chains are sized by the contact. So residues that make large contact are thicker than those that make a smaller contact. So you can see with our lysine here, it's making clear contact with the chain, the green chain C, and the yellow chain, which is the B chain. Okay. So now we know which chains interact. This helps us for the protein-protein interaction prediction. But in most cases, maybe you just have two chains, and so it's less complicated as this. But so we go to lysine 15, which is the residue that we want to mutate. And you select it here, and then you go to mole mechanics, to the try mutation. And the next option down is protein-protein binding. So prediction of the effect of the mutation on protein-protein binding here. And when you click that option, you will get a similar dialog box as before. Just there's a difference in the fact that we have this interacting part region here. So for the results of this, I have selected all, but you can, if you want to play around with it, you can select a smaller set, or you can choose all, or as before, you can also use unnatural amino acids shown here. So we know that the interacting part, the main part, is the D chain. That's the D chain where we made where the residue is that we're making a mutation on. So that's the interacting part one. And then the other chains we know are that interact are the orange chain, which is chain D, and the green chain, which is chain C. So you need to put here C and then comma D. Sorry, B comma C, B comma C. So the B and C chain, they interact. So in the results, you can see the particular energy terms related to the prediction. If you're interested in how the score is calculated, you can click here. And then we just press OK here, and it will run the simulation. Okay. I'm just going to show you the results. It takes a little for this example. I have the results of all mutation. Let's go File, Open, and have the results saved somewhere. Yeah. I cancel that. So so when the simulation is finished, you get a similar table as before. Excuse me. And so you can double click on each row. You can rank it by delta delta G of binding or delta delta G of stability. So as I mentioned, less than 0 would be a stable mutation, and greater than 0 would be would cause protein instability or less favorable mutation for stability. So we can color code this column. For example, you can sort the column by delta delta G simply by clicking in the column header here. That will reorder them. So we have in the table we have the name of the protein here, the chain where we made the mutation. We have the wild type residue, the residue number, the residue is mutated to, and it just shows that mutation. And then you have delta delta G of binding, stability, and solubility. So we may be interested in those that have low delta delta G binding as well as stability. So you could make a plot of those two columns, or actually first we're going to just color code the column. If you right click on any column, you can just right click and choose format color action here, and we can it just makes it easier to see. You can choose rainbow and it will color that column, or we can just color the other one as well. Okay, so it'd be nice to maybe look at those with good stability and binding. So you can make a plot. If you select those two columns, there's an option to make a plot here in the table. Just click on that, and that will plot. Made a sorry, made a bar chart. I don't need to just do plot. Okay. You can also maybe color those by solubility if you wanted to. Just like this. This will help us find the ones in the table that have good delta delta G of binding and stability. So the best one is obviously the wild type, which is lysine to lysine. There's another one here, which is lysine to arginine. If you're interested in looking at that one, you just double click on it and it will load it into the structure shown here. And when you load it in, you also get another, each time you click on this table, it loads the mutation. You get a new object in the ICM workspace here with the mutation. So you could save that as a PDB file or an ICM object. Use it in another program if you wanted to. Yeah, so this is the results table for the protein-protein interaction. So the process is similar for peptides as well as small molecule drugs. The only difference is that there's options for in the dialog box for the peptide, for example. You can try every single residue on the peptide and rank mutations based on delta G of binding, the effect of that mutation on peptide binding. And also, if there's a small molecule, you can try every single residue in the binding pocket with different mutations as well. So just practically how to do that in ICM. You would read in the example that you're interested in. We'll go back to that original one, the peptide bound to the PDZ domain on TP5. PDB search. And then it's the same procedure, so it's mole mechanics, try mutation, and then you have here the options for peptide binding and ligand binding. So you can go to peptide binding, for example. You can scan the peptide sequence. So you can choose, you could try some alanines at each point, or you could try all of them at each point in the peptide, and then just go OK. I don't have a selection made, so it's not activated. Or you can actually instead do, let's make a selection so it becomes, or you can choose the option scan pocket surface, and that will make the mutation at each point in the interaction site with the peptide. So it will try all the residues in this region here. And for small molecule, it's the same procedure. Just try mutation and then protein ligand binding. You can choose the option scan pocket surface if you want to try mutations all the way around the ligand binding site. This keep all chains option is in case you've got multiple chains, it will delete them if you don't choose that option. So, okay, so those are the main options for predicting mutations. So I want to thank you very much for your attention. If you have any questions, please email or call. Next week's webinar is on ligand guided modeling using known ligand binding data to mold your binding pocket for small molecule drug discovery. And we're also holding a workshop in San Diego in California, April 16th, as I mentioned before. It's free. You just sign up online. But this is actually, it's not a webinar, it's actually physically in San Diego. So you need to be here around in this area. I've got a few questions I'll try to answer.